My name is Nick Altemose. I'll be starting my group at Stanford in the Department of Genetics this July. So for the last 20 years, about 8% of the human genome has been entirely missing from our reference assembly. And that's not because these sequences are unimportant, it's because they were too difficult to sequence given their repetitive nature. And in fact, we know that these regions are really important. They tend to fall around the centromeres and pericentromeres and rDNA loci on each chromosome, which we know are important for nuclear organization, cell division, and genome stability. And just recently, some long-read sequencing technologies have matured in a way that's allowed us to assemble across these regions for the first time, such that this year we were able to release, as part of the telomere to telomere consortium, the first truly complete sequence of a human genome, including all of these formerly missing regions. And now that we have their sequences, and now that we can begin to routinely assemble them in many individuals and across different species, they're open for exploration in a way that they haven't been before. But to do this, we need to develop new tools and technologies that will allow us to overcome some of the challenges of working with repetitive DNA. And towards that end, during my PhD with Aaron Streets, I helped develop a new technology called DMELO-seq, which allows us to use long-read sequencing to map where proteins in DNA interact, simultaneously with DNA methylation information. So we can begin to probe the organization and the function of these repetitive regions for the very first time. And in doing so, what we really want to understand is how these regions are regulated and how they evolve. How does their sequence and their sequence variation relate to their function in the cell? So if you're interested in exploring some of these new regions of the genome with me, or if you're interested in collaborating on single molecule sequencing or mapping protein DNA interactions, I'd love to talk. Thank you.